sir. Yes, sir.
तर आजच्या मीटिंगला सुरुवात करण्यासाठी आजचा चांगला दिवस आहे. तो दिवस म्हणजे आज जागतिक महिला दिनाचा. सगळ्यांना शुभेच्छा. त्यासाठी मी एक वर्ड वाचून दाखवतो. आयुष्याच्या प्रत्येक टप्प्यावर आपली योग्य भूमिका बजावून यशस्वीपणे आपले कर्तव्य जगाला दाखवून देणाऱ्या आई, बहीण, पत्नी, मुलगी, मैत्रीण अशा विविध रूपात पुरुषाच्या मागे खंबीरपणे उभ्या राहणाऱ्या समस्त स्त्री शक्तीला जागतिक महिला दिनाच्या हार्दिक शुभेच्छा. नमस्कार सर.
नमस्कार. नमस्कार. तुम्हालाही सगळ्यांना हार्दिक हार्दिक शुभेच्छा.
महिला दिनाच्या परत शुभेच्छा.
गुड आफ्टरनून सर. येस येस गुड आफ्टरनून.
गुड आफ्टरनून शिंदे सर. गुड आफ्टरनून. I welcome you all for online viva of my student नामदेव पवार. एक कोटेशन वाचतो मी थोडस. It is a true that every effort is not converted into success, but it is equally true that success does not come without effort. Okay. It is my immense pleasure to introduce to today's dynamic personality with us, Chairperson Dr. Rajendra Sindhe, Principal of St. Xavier College, Autonomous Mumbai, and another dynamic personalities with us, examiner, external referee, Professor R.M. Molani sir, retired professor and director, School of Life Science, Swami Ramanand Tirth Marathwada University, and I am Dr. Suryavanshi, research guide and internal referee. Today we are conducting the open defense and open viva of my student Namdev Pawar. Today's date 8 March 2024. Time is 4 p.m. Subject botany. Student of today's viva Pawar Namdev Bapurao Pira, and research guide external referee myself Dr. Suryavanshi. The today's topic of presentation, application of biofertilizer and seed health management of paddy Oryza sativa in Maharashtra. Okay. Could we start the presentation, sir?
Yes, sir. I request the chairman Sindhe sir to start the process. No problem.
One more question, sir. हे झूम मीटिंग वरती 40 मिनिटाचं हे प्रेझेंटेशन आहे सर, पण आता जॉईन व्हायला 10 मिनिटं गेले.
You can finish it fast.
Okay sir. Okay.
Candidate can finish it fast.
Okay sir.
Sindhe sir can decide it.
Okay sir. Okay.
Yes.
Okay.
Sir, he can finish it fast. If not, sir, you should relog again, and start it again.
Yes.
Yeah, after 40 minutes, you can do it again.
Okay.
Yes, no problem. No problem.
Yeah. Again, rejoin after 10 minutes. Okay, no problem.
Yes, no problem.
Okay, please.
Best luck to the candidate.
Yeah. Where is the candidate? But
good afternoon to all of you. Myself, NB Power, working as the head and associate professor of the Vati MPSC College, Panvel. I am presenting the Ph.D. viva on the application of biofertilizers and seed health management of paddy, that is Oryza sativa in Maharashtra. My research work is divided into two parts. First part is related with the application of the biofertilizer, and second part is related with the seed health and management. Here, you know that rice is a staple food for over half of the world's population. It is a primary source of the energy, that is carbohydrate, for over the half of the world's people. It contains decent amount of fibers, protein, vitamins, magnesium, manganese, and iron. India being the second largest producer and consumer of rice in the world after China. In India, the area of production of the rice is 43.27 million hectares.
I request all
Kavita
of the
Kode-
others,
Kav-
they can mute their microphone. They can mute their mobile.
ओके सर
कविता कोणे म्यूट करा.
म्यूट कविता म्यूट करायचं बाकीच्यांना म्यूट आपण
Yes.
ओके सर
Application. Very good.
पवार
Yes,
सर
go
सुरू
ahead.
करा.
ओके सर थँक्यू अच्छा
Different types of the chemical fertilizers are used for increase the productivity of the plant, but there is hazardous effect which takes place on the soil. Leaching of the soil takes place, polluting the water basin, destruction of the microorganisms which are present in the soil, they are also destructed, increasing the susceptibility of the crop, reducing the soil fertility, soil nutrient depletion, resulting the agricultural yield decline. So for that purpose, organic farming is essential. It is application of biofertilizer is actually part of the organic fertilizer. In such circumstances, comprehensive data on the use of the biofertilizer and seed health management of the paddy is essential. So it is found that different types of the biofertilizer, living organisms, they can help increasing the yield without damage associated with the chemical fertilizers. So biofertilizers are small microorganisms which are responsible for enhancing the absorption of the nutrients which are already present into the soil, but they may be in inorganic form. They may be converted with the help of the synthesis of the different types of the reaction. These biofertilizers are generally applied on the seed surface, or they may be applied directly in the soil. They are renewable, sustainable, and inexpensive. That is eco-friendly, and it is a main alternative to the chemical fertilizer for growing the plants. This biological activity of the specific organism which acts as a biofertilizer, they may be nitrogen fixers, phosphate solubilizer, mobilizer, biofertilizers for the micronutrients, growth promoters to help improve the soil fertility by fixing the atmospheric nitrogen. These biofertilizers are also responsible for secreting the growth substances and some antifungal chemicals which are responsible for improving the germination and the root length. Ultimately, productivity of the plant will be decreased. These biofertilizers also increase the soil structure and texture of the soil may be due to biofertilizers. So they are most essential. In the first part, in the second part, seed is also most important because this seed is must be healthy. But different types of these seeds, they are also covered with the help of different pathogens. They act physically, physiologically, and total health of the seed will be restricted, and loss of the germination percentage will be takes place, and ultimately plant productivity will be decreases. Mostly seed carries the fungi, bacteria, nematode, viruses, which are directly present on the seed, which lost the crop yield. Diverse array of the pathogen, including the microorganism, nematodes, viruses, mycoplasma are responsible for the rice plant diseases. The most significant of these fungi causes the blast of rice, that is caused due to the pyracularia, then brown spots. Total about the 13 genera of the fungi were found to be associated with the seed sample of the rice.
Yes, Pawar sir, go ahead.
Of my research, collection of the rice seed sample from the different locality of the Maharashtra, the screening of the mycoplasma using the different methods, the bacteria associated with the seeds of the rice cultivars, effect of the dominant fungi on the seed health of the rice, collection of the biofertilizer from the various research institute for the treatment of the rice rice crop as the bioinoculum. Use of the biofertilizer for treatment on the rice crop for increase the productivity as compared to the control. Physiological and pathological changes which are takes place in the rice. Effect of the fungicide seed treatment on the seed microflora, seed germination and vigor index. Effect of the antagonist on the seed microflora, seed germination and vigor index. Effect of the plant extract on the seed microflora, seed germination and vigor index. Seed-borne pathogens of the Ph.D. using the fungicides, microbial agents, material and method. This slide shows the location map of the experimental site, that is a research field of the MPSC College Panvel, as well as a laboratory of the KV College, Pendarkur College. In the first part, I already stated the effect of the biofertilizers on the germination and various growth stages. For that purpose, seeds are collected from the Kharland Research Station, Dr. Babasaheb Sawant, Konkan Krishi Vidyapeeth, Panvel, District Raigad. Then collection of the different biofertilizers like azospirillum, bacillus, blue-green algae, mycorrhizae, pseudomonas, trichoderma, and for the comparison, one chemical fertilizer is taken, that is 1990 as to 90. Healthy seeds are separated from the destructive seeds with the help of the seed upgradation technique. For that purpose, salt is used. The specific gravity is 1.3 specific gravity water is taken for the separation of the healthy seeds. So in this healthy seeds are experimented further. Combination of the biofertilizer which is done for the different various research purpose for the germination as well as for different treatment. T0, that is control, T1 chemical fertilizer, T2 blue-green algae, T3 azospirillum, T4 bacillus, T5 trichoderma, T6 mycorrhizae, T7 fluorescence. They applied singly, and in the developed treatment, blue-green algae plus pseudomonas, blue-green algae plus mycorrhiza, azospirillum plus bacillus, and in the triple inoculation, azospirillum, bacillus and pseudomonas, they are applied, mixed together and applied on the seed or directly given to the plant. I have studied this work, effect of the biofertilizer in from the seed germination up to the productivity of the plant. In the first stage, effect of the biofertilizer on the seed germination and seedling growth of the paddy. For that purpose, the seeds are inoculated singly, dual, or they are combined together. And after 7 days, germination percentage, root length and shoot length, fresh weight and dry weight, and vigor index is studied with the help of standard method.
Second.
In the first part, seed treatment is given in the pot as well as into the soil. But for the seed treatment, effect of the biofertilizer on the seedling growth up to the 1 month is also studied at interval of 10 days, 20 days, and 30 days. Same parameter, morphological and some physiological parameters are also studied. Then in the seed treatment, vegetative stage, what is the effect of the vegetative stage? It is studied with the help of morphological parameter, that is shoot length, root length, fresh weight, dry weight, physiological parameter, that is photosynthetic pigment. Then physiological parameters, estimation of the total carbohydrate, estimation of the total nitrogen and protein, estimation of the protein, estimation of the proline, and estimation of the polyphenols. All these parameters are studied. This slide shows second method, that is the seedling root dip method. For that purpose, 21 days old seedlings are transplanted into this bed. And same parameters, that is morphological and physiological parameters, are studied at in the next third method, it is most important. There 2-year experiment, that is a field experiment is done. For that purpose, research, the bed preparation is done in the RBD method for the two Kharif seasons. Effect of the biofertilizer on the soil as well as on the plant is studied. For that purpose, climatic data, climate and weather data is taken from the Meteorological Observatory, Agriculture Meteorology Division, Pune Subdivision, Panvel. Then soil analysis before experiment is done in the soil survey and soil testing laboratory, Thane, Maharashtra. This is the RBD design. Here, total 36 subplots, one by one subplots are arranged in triplicates. Before that, these are soil parameters that is studied before the treatment. Here combination, that is standard dose of this biofertilizer is given to the particular that is a 36 plot, that is a triplicate. Here crop which is taken for that is a Jaya variety. Then plot size 1 by 1 meter, number of plots 36 net experimental plot, that is a date of the sowing. So total number of treatments are 12. This is actually layout plan of the RBD method with the triplicates. During the experiment, before the starting of the experiment, nursery plants are cultivated or nurseries are developed in the nearby farmer. This nursery plot of the paddy for the 2 year. For the use of the 21st days old seedling, this nursery is used for 2 Kharif season. This slide shows the life cycle of the plant. Their analysis of the soil at the pre-sowing position as well as post-harvest stages take place, and growth parameters of the paddy were studied at 30 days, 60 days, 90 days after transplantation during the harvesting. So harvesting and threshing is also done at the end. Different parameters are studied at this interval, that is height of the plant, number of the leaves per hill, then the total number of the tillers per hill, dry matter production per hill, yield parameters, that is length of the panicle, weight of the panicle, then spikelet per panicle number, then number of the field grains per panicle, test weight, then grain and straw yield per kilogram per hill or quintal per hectare, and biological yield and harvest index. Physiological parameter for all the intervals, that is the 13, 16, 90, and during the harvesting, physiological parameters only from the leaves are done. Chlorophyll, total carbohydrate, total nitrogen and crude protein, total nitrogen, then total protein, proline, polyphenols. For that purpose, preparation of the acid digestion takes place. For a study of the micronutrients, macro and micronutrients, that is major and minor elements are also studied. Major elements nitrogen, phosphorus, potassium, calcium, and magnesium. Minor elements which are studied in this experiment, they are iron, manganese, zinc, and copper. From the grain, total carbohydrates, total nitrogen, total protein, proline, macronutrients and micronutrients are also studied, and estimation of the phosphorus is done with the second method and EDT method. In the second part, for the seed health management, here collection of the seed takes place from the different 70 species are collected from the different regions, that is North Konkan, South Konkan, North Central Maharashtra, South Central Maharashtra, Marathwada, and Vidarbha. Out of the 70 varieties, only 10 varieties are selected, randomly selected for the
Next.
microflora study. Screening of the rice cultivar for rice cultivar. For that purpose, only 10 rice cultivars from the different locality were selected for the screening of the microflora. Here, visual examination of the seed from the storage is done. Then isolation of the seed microflora is done with the help of the agar plate method, blotter method, and seed or seed method. And identification of the fungi is done and identified with the help of standard identification of manure of the soil fungi. Next. Physiological changes which are takes place within this storage seed, their total carbohydrate, total nitrogen, total protein, proline, polyphenol, mineral, nutrients, and potassium. Then physiological parameters are studied. Here, effect of the fungicide, the seed treatment of the seed microflora for that for studying the seed germination and vigor index, carbamazepine, copper, oxychloride, thiram, mancozeb, captafol. For study the effect of the antagonistic treatment on the seed microflora, seed germination, and vigor index, biological member, that is a trichoderma, two species of the trichoderma artianism, trichoderma viridis. Then bacteria, that is Pseudomonas fluorescens and Bacillus subtilis is used. Effect of the biological control on the seed-borne pathogen using the antagonist, the same, that is a trichoderma and Pseudomonas and Bacillus species used for the plant extract. Effect of the extract, that is Azadirachta indica, Vinca rosea asimum, then Polythia, Tridex, Phytex. These locally observed plants, local plants are used. If you show the experimental result of the entire this work, that is the most important finding. Effect of the biofertilizer on the germination percentage on after 7 days old seedling of the paddy. Here, rate of the germination is increases in the combined form, that is mixture of the azospirillum, bacillus, and Pseudomonas. Then seedling length of the seedling, fresh weight, dry weight, and vigor index is also increases due to the combined effect of this biofertilizer, as well as shoot length during this 10 days interval, 20 days interval, and 30 days for 1 month study of this seedling. Here, seedling growth, stimulation of the seedling growth takes place due to the again combined biofertilizer, which are compared with the control and chemical dose. Then growth parameters, shoot length, root length, fresh weight, dry weight, then photosynthetic pigment, that is chlorophyll A, chlorophyll B, chlorophyll, total chlorophyll. Then in the seed treatment, this slide shows the effect of the seed treatment. Here, morphological parameters, shoot length, root length, fresh weight, dry weight. Here, combined form, that is a T11 treatment, azospirillum, bacillus, and Pseudomonas shows the best, that is highest seedling length. But double, that is a dual fertilizer is also best, shows the best growth of the rice. Then physiological parameter, that is total carbohydrate, total protein content, proline content, and phenolic compounds, they are also increased due to the stimulation of these different biofertilizers. Then if you show the effect of the biofertilizer on the growth of this Ph.D. due to the seed treatment, root length as compared to the control root length, then shoot length, root length, shoot length, as well as fresh weight and dry weight is increases due to the application, combined application of the biofertilizers. Then physiological parameters, photosynthetic pigment, except chlorophyll B, chlorophyll A, and total chlorophyll will be increases due to the effect of these biofertilizers. Then total carbohydrate content will be increases due to the combined effect that is a T11, azospirillum, bacillus, megatherium, and pseudomonas. Then physiological parameters, that is protein content, they are also stimulated due to the addition of the single or double as well as a triple application of these biofertilizers. Then proline content, generally prolines are synthesized during the drought condition, but due to this application of the pseudomonas and due to the mycorrhiza and trichoderma, here increase in the proline content takes place. Then polyphenol content, it is also responsible for increase the drought condition of the plant. Here, if you see the result at the TJ, that is azospirillum and bacillus, it shows the best effect on this increasing the phenomenal content. Then this slide shows the second method, that is the seedling root dip method. For the seedling root dip method, 21 old days, seedlings are dipped into the root in the different biofertilizer treatment and they are sown. And I have also observed that in all the parameters or in all the treatments, that is from blue-green algae up to the triple treatment, azospirillum and bacillus, it is most effective for the entire plant growth with respect to their parameters. It may be morphological parameters which was studied at the 13, 16 and 90 days. That is shoot length, root length, fresh weight and dry weight. Physiological parameters are same, photosynthetic and other. In case of shoot length, here BJ and mycorrhiza shows the highest effect on the shoot length of this plant. Then root length. Root length is also increases. It increases at the pseudomonas fluorescent due to the application of this root length will be increases. Then other parameters, fresh weight, dry weight, and physiological parameters like chlorophyll A content, there is no major change into the chlorophyll B content, but total chlorophylls are increases. Then physiologically, these chlorophyll pigments, that is the synthesis of the chlorophyll pigment takes place due to the addition of some chemicals. Here, total carbohydrate protein contents are increased. It gives the best influence of this biofertilizer on this total carbohydrates. Then proline and polyphenol. If you see the influence of this biofertilizer on the proline and polyphenol content in the leaves, it is added. So, for the combination, that is the influence of the biofertilizer treatment on the successive stages of the growth. Here, azospirillum, bacillus, pseudomonas, and trichoderma gives the best effect on this method. But for all this data of the 2 years, that is pooled data of the 2 years, with respect to growth parameters like height of the plant and number of the tiller, the number of the leaves, dry matter production, physiological parameters, that is photosynthetic pigment, total carbohydrate, then total nitrogen, crude protein, proline, polyphenol content, micronutrient and macronutrient, then yield parameter, length of the panicle, weight of the panicle, number of the spikelets per panicle, number of the field grains per panicle, test weight, grain and straw yield per hill, grain and straw yield quintal per hectare, biological yield and harvest index, then these biofertilizers also affect on the composition of the grain. That is the total carbohydrate and protein content into the paddy grain. That is the Jaya variety. The macronutrient and micronutrient into the.
Excuse me, everyone. As meeting is going to end within the few seconds, so I would like to request the chairman, examiner, both examiners and the participants to join after 10 minutes.
Are you
Yeah.
sending the separate link or the same link?
I think same link will be there.
Okay, okay, fine.
Yeah.
Yeah, okay, continue.
Yeah.
As a
Okay,
minute,
sir.
yeah.
Continue. Hello, Gati, link.
This is a 2-year experiment. Here, I have studied the influence of the biofertilizer treatment of the successive growth stages of the paddy. Of the
आवाज येतोय ना, sir?
येतो
yes.
चालू दे.
Okay, sir. Thank you. Sir. Continue.
Yes.
here in the entire growth parameters, if you see the influence of the biofertilizer treatment on the successive growth stages of the paddy, here again combined effect as well as individual or single. Where actually for this field 2 doses are given. First dose is given at before the 13 days, and after that during the tillering stage. After tillering stage, this effect, this is the most effective results are observed. This slide shows the effect of the different biofertilizers on the paddy variety at 60 days after transplantation by this RBD method. Actually, guide is also visited this field. Then these are the different stages. That is the tillering stage. Then this is the panicle stage, flowering stage, and during the before harvesting, length of the panicle and other parameters are also calculated. Here, during this experimental days, that is the pooled data is taken for the calculations. Is average rainfall, climatic condition, all the data is taken from the Panvel. Their average rainfall, temperature, maximum, minimum, rainy days, relative humidity, wind speed. That is, all these climatic conditions are also affect, these environmental conditions are also affected the entire life cycle of this paddy. Here, growth parameter, that is the height of the plant, number of the tiller per hill, number of the leaves. Then dry matter production is increases due to the application of the single, dual or triple uraculum. Then physiological parameter, that is photosynthetic pigment, that is chlorophyll A, chlorophyll B, and total chlorophyll. Total carbohydrate, total nitrogen content, fluid protein content, proline, polyphenol, then micro and macronutrients yield contributing parameters are also studied. Here, these biofertilizers singly or doubly or triple form of the biofertilizer, length of the panicle, weight of the panicle, number of the spikelets per panicle, number of the field grains per panicle, test weight is also increases, then straw yield per panicle is also increases, and grain, quality of the grain with respect to physiological parameter, that is total carbohydrate content, then micronutrients and macronutrients are also studied, and it shows a good effect of the biofertilizer, and highest effect of this biofertilizer takes place on the grain production. Here, this graph shows effect of the biofertilizer on the plant height, then number of tillers, then effect of the biofertilizer, number of the leaves. In case of this factor, number of leaves are increases up to the 90 days. After that, there is no change. There is stable. That is stable or constant number. There is no other leaf formation done. Then dry matter production is also increases. In the case of yield parameter, length of the panicle is generally increases at T10. Azospirillum bacillus megatherium and highest length of the panicle is observed in the treatment of the Azospirillum bacillus and Pseudomonas. Weight of the panicle is also increases with the dose, that is with the application of the single, double and triple biofertilizer treatment. Weight of the panicle is also increases. Here, number of the spikelets, they are increased and they are actually as compared to the chemical fertilizer as well as with the control. Then number of the field grains per panicle is also increases. Test weight remains constant in the T11, T10, T9, and T8, that is the megatherium and Pseudomonas. Here, test weight will be increases. That is, it is good for the growth parameters. Then grain and straw weight yield, I have calculated gram per hill as well as quintal per hectare with the help of calculation. It is also increases with this biofertilizer treatment. Then biological yield and harvester index is also calculated on the basis of the straw yield as well as grain yield. Then physiological parameters for the study of the physiology, that is for the study of the effect of the biofertilizer, PhD leaves are selected at different stages. There is 30 days, then 60 days and 90 days and harvesting. At this all the 4 stages, it is observed that rate of the photosynthetic pigment will be increases. I observe it is increases, but during the harvesting, this synthesis of the chlorophyll pigment is decreases. That is chlorophyll A, chlorophyll B and chlorophyll, total chlorophyll. Then carbohydrate content, total nitrogen and crude protein, then prolyl and polyphenol content. It is also increases when these leaves are studied with respect to macro. We know that all these parameters, that is micronutrients and macronutrients, generally phospholytic fungi, then these are responsible for the absorption of the nutrient. Major nutrients are absorbed due to this biofertilizers. And this, if you see the intake capacity or intake of this nutrient, that is the nitrogen, phosphorus, calcium, magnesium, they are increased. Micronutrients like Fe, Mn, Zn and Cu, only in the one plot, only arsenic compound is observed in only one plot. Then physiological parameters which are related with the paddy grain, that is total carbohydrate and proteins, they are also increases. Then NPK, CAMG, they are also released because due to the stimulation or due to the enzymes which are secreted by these micronutrients, that is Fe, Mn, Zn and Cu, they are also increases in the grain of the paddy. After harvesting, chemical parameters of the soil after harvesting, they are also studied. It indicates that the parameters, that is the pH, electric conductivity, organic matter, and other parameters are increased. That is, soil become rich. That is improvement of the soil takes place due to the biofertilizer application. In the second part, for the study of the seed health and management of the paddy, different seeds are taken and fungi which are associated with the seeds of the various paddy cultivars are also studied. Here, seed microflora of the paddy observe different 13 species, that is aspergillus flavor, Asparagus niger, then Fusarium algisporium, Pyracularia origi, Bipolaris origi, Alternaria, Curvularia, then Cercospora, then Rhizotonia, Sclerotia, Ephelis, and Saracladium. These are the major pathogens they are observed on the seed. Here, some visible effects are also observed. Pathological effects or changes are observed in the paddy rice. That is, seed coloration takes place in some seed. In some seed, light to dark brown dots observed. Dark purple discoloration, light pink discoloration, eye sector spot with the ash gray is also observed. Due to this seed rotting, seedling blight and rate of seed germination will be increased. Here, I have already studied the effect of the fungicide treatment on the seed microbiota, then seed germination and weak hair. Here, fungicides which are used for the treatment were Thyram, Carbendazim, Mancozeb, copper oxychloride, Capton and Captopol, ranging from 0.1 to 0.5% concentration. Among all these, only carbendazim is most effective, which is responsible for the inhibition to reduce the percentage of the seed microflora by increase the percentage and seed elimination and figure index significantly. If we show the effect of this biological control of the seed pathogen using the antagonistic like trichoderma, then pseudomonas were found to be more prominent in the controlling the seed-borne pathogens, particularly controlling the pyracularia. Other biocontrol agents are also show increased effect on the control of the fungi. Then evaluation of the antagonist are the 5 control agents. It was recorded that trichoderma, two species of the trichoderma, single species of the trichoderma and one bacteria, that the pseudomonas bacteria were found to be more prominent in controlling the seed-borne pathogen, particularly pyracularia. So, antagonist seed treatment on the seed microbial germination and vigor index. For that purpose, different biocontrol agents are used. That is a trichoderma, 2 species of the trichoderma, then pseudomonas and bacillus bacteria are used. Here, trichoderma, Hergianum and Pseudomonas fluorescens shows the best observation with related to the germination treatment resulting in the greatest degrees into fungal connections. Here, effect of the plant, effect of the plant extract on the seed microflora. Here, effect of the different plant extract on the percent seed microflora, seed germination, and vigor index studied. Extract of the agile director indicates that is enzyme was more effective as compared with the other plant extract. Seed germination was steadily rise as the plant extract doses. Effect of the chemical control of the seed-borne pathogen using the fungicide. Then, if you see the entire summary and conclusion, here we know that this rice is a dietary stable primary source of the income, and due to the excess of chemical fertilizer and due to the impact on the public and environmental health due to this pollution, use of the biofertilizer is alternative form. It is beneficial to the environment, soil quality and crop yield. So this impact is the most important on the different growth stages. Here, to investigate the entire impact on this treatment of the biofertilizer, the entire experience was divided into 3 methods, that is seed treatment method, seedling root dip method, and field application, that is RBD method. In all these treatment, that is from the germination up to the harvesting, it may be germination stage or seedling stage, it may be vegetative stage or reproductive stage. During the germination, here germination percentage, root length and shoot length, fresh weight and dry weight, figure index, morphological parameters, all these are studied with the interval of the 30, 60, 90 days and at the harvesting, in all these parameters, it is observed that these biofertilizers may be effective single A or double A or in the triple A, that is 811 azospirillum, that is mixture of these three members are responsible for the increase in the plant productivity during the seed treatment method and seed root deep method. Here, in all the parameters for these combined treatments and into the pre-harvesting and post-harvesting experiment, it is observed that single azospirillum, bacillus and trichoderma or mycorrhiza Pseudomonas, when it is singly applied to the seed or to the field, they give the best effect. But most important effect or highest parameters which are increases due to the T11, that is mixture. Here in the RBD method, chlorophyll pigment, total carbohydrate, that is all the physiological parameters which are related with the leaves. Then all the parameters which are related with the grain, that is after harvesting, they are also gives best result due to the application of these biofertilizers. After harvesting, I have collected the soil with the help of the wooden stick. And all the chemical parameters of the soil after harvesting, they are tested into the laboratory. And it is also observed that improvement in the soil takes place. pH remains constant, electrical conductivity remains constant, organic matter, organic carbon will be increases due to the application. Total nitrogen, available nitrogen, that is total nitrogen, total phosphorus and potassium will be increases due to the effect of this biofertilizer. Due to this, in the case of the seed health and management, I have studied that I have used the three methods: the agar plate method, blotter paper method, seed wastage method. For assessing the percentage incidence of the fungi, the agar plate method was superior than the blotter seed wash method. All the cultivars were screened for the seed under the unsterilized condition. 10 cultivars of the rice with Basmati, Jaya, Komal, Panvel 2, Priyanka, Rajni, Ratna, Sai 212, then Samba Masuri and Vaishnavi. All these species of this rice variety indicates that is 13 species of the fungi which are assessed from these 10 varieties. Then the cultivars of the rice are associated with the dominant fungi such as Pyracularia, Fusarium, Alternaria, Aspergillus, and there are two types of the Aspergillus niger and Aspergillus flavus, causes the seed discoloration, seed rotting, and seedling blight and seed rot. Effect of the different fungicide was observed on the seed microflora, seed germination, and vigor index of the rice. Here, carbendazim is the best. It is found that it is the most inhibitorial to reduce the percent of seed microflora. Here, seed treatment by the different biocontrol agent, that is trichoderma, two species of the trichoderma, Pseudomonas fluorescens, and bacillus. Among these, trichoderma Hergianum and Pseudomonas fluorescens were most effective. Here, effect of the different plant extract on the percent of the seed microflora, that is the seed germination, vigor index also studied. Leaf extract of the neem, that is Azadirachta indica, was more effective as compared to the other leaf extract to reduce the seed microflora and increase the seed germination and vigor index, as well as the effect of the different biocontrol agents, that is antagonist, was studied against the Pyracularia. By dual culture method, the different biocontrol agents used was Trichoderma hergianum, Trichoderma viridis, and Pseudomonas fluorescens and Bacillus. Trichoderma hergianum and Pseudomonas shows a more inhibitory effect on the cerebellum. During the entire course of the research, I have published different we have published different research papers. This is list. And finally, I wish to extend my cordial thanks to chairperson and principal Dr. Rajendra Sinde, external referee Professor Ramjan Mulani sir. I am truly grateful. Thanks to my research guide Professor Nilkant Survensi, Department of Botany, for his valuable guidance, continuous motivation, and constant support. I am extremely thankful to Honorable Dr. Ganesh Thakur, former Secretary Race Section South Satara and Principal of the Mahatma Phule College. I am also thankful to all my colleagues. I am deeply invited to all the members. Thank you, sir.
Thank you, sir. Sinde sir. The topic is open for discussion.
Yes,
Sinde
Mulani
sir.
sir.
Questioning. Okay, sir.
Mulani sir,
Okay.
the external examiner
Okay.
will ask
Okay.
questions first.
Mhm.
Once he finishes, then we will
I
open
request-
it for public. Mulani sir, please go
First-
ahead.
Okay. The topic is open for questioning. Any question from participant? Audience?
Mulani sir
Whether
विचारतील
I am
पहिल्यांदी.
audible?
Mulani sir, please.
माझा आवाज येतो का?
Okay.
हा
येतो,
येतोय
येतो.
सर बोला.
Yes,
येतो,
Pawar sir.
हा,
Please
बोला.
tell me what is the basis for selection of those plants for giving treatment? Few plants you have selected. What is the basis for selection of the plants? Because you have selected different plants.
This sir, they show the microbial and microfungal activity.
Okay.
Generally, they
Okay.
are used for the
Okay. And then other
microfungal
thing is,
treatment.
I have not a question but suggestion, because in physiological parameters there are three additional LAR, NAR and RGR, because you are having data for 3 times 30, 60, 90. Okay. So if you use these values, because you are having already values with you, if you do,
Yes.
you can do excellent publication through your observations. Okay. And you have done a good, excellent work, both, experimental work as well as your presentation of thesis and presentation, the PowerPoint presentation was very good. So from my side, I congratulate you for giving excellent presentation. And from my side, I can suggest, recommend the chairman to recommend the university for award of you as a Ph.D. degree from the University of Mumbai. And I'll also congratulate the guide for excellent work taken from the student. So it is very nice, very good. Sinde sir, from my side, it is very clear.
Okay, so if there are any questions from the
Thank you, sir.
participants,
Okay, sir. Any question from
Participant?
participant? Please.
sir, your climatic data, the climate related data is
Yes, yes.
2014, 2016. त्याच्यानंतर तुम्ही परत डेटा घेतलेला नाही का त्यांच्याकडनं? फ्रेश डेटा?
नाही, आपला एक्सपिरियन्स सर 2च वर्ष होता आणि 2 वर्षाचा एक्सपिरियन्स आपल्याला 2 खरीप सीझनचा डेटा पाहिजे होता आपल्याला.
नाही, पण
आता आपल्या
कधी
एक्सपिरियन्सच्या
सबमिट
सीझन
थिसिस कधी सबमिट केला हा?
थेसिस आता सबमिट केला. काय कारणास्तव लेट
एक
झालं
वर्ष
काय?
झालं सर thesis. एक
एक
वर्ष झालं.
वर्षाला
हा.
सबमिट
एक
करून.
वर्ष झालं.
ऍक्च्युली
Okay.
वर्क आहे ते 13, 14, 15, 16चा आहे.
फील्ड वर्क
अच्छा.
13, 14, 15.
ओके,
Okay.
ठीक आहे. काय बाकी कोणाचं क्वेश्चन नसतील तर आपण
Okay. Participant
सो
and all my student, can you ask any question?
Time bagawa, Suryavanshi sir, you can take time.
Okay, okay, sir. Okay, thank you.
Hello, sir.
Hey, sir, I.
विचारा लक्ष्मण राठोड.
Okay, थांबा. क्वेश्चन आहे का कुणाकडे?
No, sir.
Participant.
No, sir. No, sir.
Okay.
Okay then.
Over to Rathore sir.
Yes.
हा.
May I audible, sir?
Oh, bola bola
yes.
काय.
Okay.
Last part of the presentation, vote of thanks, please go ahead. Give permission of research, external referee and Sindhu sir. Sir,
Yes, yes, yes.
I would like to congratulate as a chairperson of this Viva, the congratulations to student Dr. Pawar. Let us call him doctor now. So Dr.
Yes, yes.
Pawar and the guide Nilkant Suryavanshi sir. Congratulations to your excellent work presented today. And thank you, Mulani sir, on behalf of University of Mumbai.
Thank
I
you,
would
sir.
like to
I'm thankful
officially,
to the authority for
yes,
giving me an opportunity.
yeah,
Okay,
yeah.
sir.
So congratulations once again, and we can conclude. Yeah, you can
Camera
start the.
ready,
Please go ahead. Rathod sir,
Sir, may I propose
please
vote of
give the
thanks?
vote of thanks. Okay.
Okay, sir.
Yes.
Vote of thanks, please.
A very good evening to all. On behalf of our online PhD viewers, I am glad to here express my vote of thanks. It's my privilege to propose a vote of thanks and acknowledge the contribution of those who worked really hard to make this PhD viewers. Our most valued honorable chairperson, Principal Dr. Rajendra Singh sir, Saint Xavier College, Mumbai, Professor Dr. R.M. Mulani sir, External Referee, School of Life Science, Swami Ramanathirt, Marathwada University, Nanded. Professor Dr. Nilkant Suryavanshi sir, Internal Referee as well as Research Guide, Research Center, K.V. Pendarkar College, Dombivli. Principal of K.V. Pendarkar College and Principal of Mahatma Phule ASC College, Panvel, Dr. Ganesh Thakur sir. Vice Principal R.A. Patil sir, Research Student, Professor N.B. Pawar sir, all research scholar and professor of online PhD Vivos. We would especially like to thank Chairperson and Principal Dr. Rajendra Shinde for accepting our invitation and joining us today. Your presence has boosted our energy and your thoughts have inspired us all. Thank you, sir. I would like to extend thanks to external referees, Professor R.M. Mulani sir. Sincere appreciation for taking time out of his busy schedule to attend and work as an external referee for this PhD Vios. Thank you so much, sir. I would like to thank our beloved research guide, Professor Dr. Nilkant Suryavanshi sir, for being the catalyst that stimulate us to do our best and standing as a pillar of strength with the deep sense of appreciation. We thank our loving research guide for their tireless efforts. Thank you so much, sir. We must special thank to our beloved principal K.V. Pendarkar College, Dombivli, and Principal Dr. Ganesh Thakur Sahib, Mahatma Phule ASC College, Panvel. Valuable cooperation during research work. Finally, I would like to thank all of you present here for making the time to be with us.